of two different kind of tospoviruses. It could be groundnut bud necrosis virus, or it could also be due to another tospovirus which is known as capsicum chlorosis virus. So that clearly shows, based on symptom alone, you cannot make out whether it is because of GBNV or whether it is because of capsicum chlorosis virus. Okay? I will give you another example. Watermelon. There is a disease in watermelon, what is known as watermelon bud necrosis disease. And when we started working on this disease, and we found that this disease is caused by a tospovirus, and that tospovirus was designated as watermelon bud necrosis virus. But subsequently we realized it is not only caused by a tospovirus, what is watermelon bud necrosis, it is also caused by another tospovirus which is common on leguminous crops, that is groundnut bud necrosis. So what I am trying to say, based on symptoms, you cannot make a precise identity unless you subject these samples to molecular diagnostics technique like ELISA or PCR. Then only you will be able to know whether that given disease is caused by which virus and so on and so forth. Like in fungal diseases or the bacterial diseases, whenever you pick up a sample, the first thing you do is you examine them under microscope. And if you are fortunate to have electron microscope for viruses, then the immediate spot check examination can be done by electron microscope. But unfortunately, if you do not have electron microscope, which is not possible in every lab, then you have to subject those samples initially to ELISA by using a panel of antibodies actually. Okay? This is just to give you an example that what is our diagnostic capability. Over a period of time, an array of diagnostic techniques have been developed actually. And for the sake of convenience, I have classified them, the techniques based on biological properties of the virus, techniques which are based on physicochemical properties of the virus, techniques which are based on immuno-based techniques, and techniques which are based on the nucleic acid. So for the sake of convenience, we have classified them into the various categories. And I would like to draw your attention to the sensitivity limit or the detection limit of the various techniques. And if you come down to this, you will find that this is such a technique wherein you can go up to attogram. Okay? So that is the kind of the sensitivity limit or detection limit we have with regards to the various diagnostic techniques which are available with us. Okay? Now, among the various diagnostic techniques which have been developed over a period of time, ELISA, which was developed in 1970s, and PCR, which was developed more recently in 1990s, these are the two techniques which are very routinely used now as far as diagnostics is concerned. It is immaterial whether we are talking of fungal diagnostics, bacterial or viral. If you go through the papers, you will find that these are the two techniques which are routinely used for fungal or for virus diagnostics. And again, among the two, ELISA and PCR, it is ELISA which can be routinely used. It does not require very sophisticated facilities. A lab with very rudimentary facilities can make use of ELISA. Okay? So among these two, ELISA can be routinely used in any diagnostic laboratory. Now, if you talk about immuno-based diagnostics, or if you talk about nucleo-based diagnostics, whether PCR or molecular hybridization, you can only know the known viruses. Suppose you want to do serology, you must have antibody. Antibody you can have only for the known viruses. Similarly, if you have to do PCR, if you have to do the molecular hybridization, you should have primers. Primers you will know only for the known viruses. But what about unknown viruses? If you really want to know the unknown viruses, there is a technique which has come now from 2009 onward. I don't know how many of you have heard this term, deep sequencing or next generation sequencing. That is the technique which can give you an idea to know the unknown viruses. Okay? You don't know which virus is, so you should subject that sample to deep sequencing or next generation sequencing. Am I clear up to this stage? Now, I will not discuss each and every technique in detail, but I will only discuss what has been the paradigm shift in the various diagnostic techniques during the last 2 decades. When you talk about serology, and if you have to use serology, you must have a diagnostic reagent in place, means antibodies, and predominantly we use polyclonal antibodies. I'll share with you that how, I don't know how many of you have the experience of raising antibody. This is the conventional method of raising the antibody. You need to multiply the virus in the glass house. Once you have multiplied the virus in the glass house, you need to mass propagation. Then you need to go for virus purification. And once you have purified the virus, this is the purified virus, then you have to immunize into the animal system. Either it could be rabbit and any other animal system. But this is a very cumbersome method. It is cumbersome. You need to maintain the virus actually. Then you require large amount of tissue. And then quality of the antibody will always differ depending upon the sample you have taken. Every time it will differ actually. Okay? So in order to overcome these difficulties, we have gone for recombinant approach actually. And that is again genome assisted actually. Okay? So on the right hand side, you could see that how do we go for recombinant approach for raising the antibody. This is just a schematic representation of a viral genome, and predominantly when we are raising antibody, we try to raise antibody against the coat protein or the capsid protein. So what we do is we try to amplify the coat protein gene by RT-PCR. Once we have amplified the coat protein gene, we try to clone the gene. I don't know how many of you have a chance to clone the gene. Can you raise the hand? How many of you have a chance to raise? You must have cloned. Which vector you are using? PGMT? PGMT. Anyway, I mean, those of you have not had a chance, maybe in future you will try to do the cloning. So first initially we try to clone the gene, as she said, right, in the PGMT vector. And once we have cloned the gene in PGMT vector, then we subsequently subclone the gene in a protein expression vector. Having expressed the protein gene in a protein expression vector, we try to express the protein and then raise the antibody by ionizing into the rabbit system. When we follow this approach, anytime you want to raise antibody, you can take the cloned gene from your fridge, go for protein expression and immunize. So you need not to multiply the virus, you need not to maintain the virus, and every time quality of the antibody may not vary that much. So this has been a paradigm shift in the recent past based on the genomics, and that's how now antibodies are raised. Just to give you a few examples, in the recent past, I mean, we have used the same approach. For example, we were working on a virus what is called as Cymbidium mosaic virus on orchids. This is an important virus when you talk of orchids. So what we did is we went for RT-PCR, we amplified the coat protein gene, cloned the gene in PGMT vector. As I said earlier, after cloning the gene in normal cloning vector, we try to clone the gene, subclone the gene in a protein expression vector, that is P-maltose system. And once we have subcloned, we try to purify the expressed protein actually. Okay? And this information has been published in 2010 by one of my associates in Journal of Phytopathology. Similarly, there is another virus what is called as odontoglossum ring spot virus, which is again a common virus affecting orchids. A similar approach we followed again for the odontoglossum ring spot virus, and we raised the antibody. And that again we published in the same journal. In the recent past, the Advanced Center for Plant Virology has raised antibodies using recombinant approach for large number of viruses, which includes those of you who work on banana. There is a virus problem, banana streak virus, Omar mosaic groundnut bug necrosis, those who work on cardamom, large cardamom chirke virus, odontoglossum papaya ring spot virus, and potato viruses. So for large number of viruses affecting the field and the horticultural crops, Advanced Center of Plant Virology, which is now led by Dr. V.K. Burnwal, I mean, that center has raised antibody by using this recombinant approach. Now, in nature, the plants suffer from multiple infection. Okay. Now, what we thought is that, like in case of when we talk of resistance, in order to incorporate multiple resistance, we try to stack the multiple genes. We thought that instead of raising antibody against just one virus, why not we raise antibody against multiple viruses as a cocktail? Then instead of using multiple ELISA every time for each virus, we can use a cocktail of the antibody, and then using a cocktail of the antibody and say whether the sample is infected or sample is not infected. Because the stakeholder, your farmer or client, is not interested whether potato is suffering from potato virus Y or potato virus M. He is concerned whether my sample is carrying the virus or my sample is not carrying. It is our academic interest that it is banana streak or it has banana bunchy top. So, in order to cut down the cost of the detection by using large number of antibodies and so on and so forth, we thought, can we go for a cocktail of the antibody? Say for example, in cucurbits, you have cucumber mosaic virus. In cucurbits, those of you who are familiar, you have papaya ring spot virus, and there is also a tosper virus. So, what we thought is, let us stack the coat protein gene of all these three different viruses, and then clone the gene, subclone in a protein expression vector, and produce a cocktail of the antibody. Now, using this cocktail of the antibody, just one set of reaction, you can say whether your given sample is having CMV, or it is not having CMV, it is not having GBNV, or it is not having PRSV. So, you are cutting down the cost actually, and that is what we did. And this information has been published recently in Journal of Virological Methods in 2014 actually. But what we thought is, this is just a proof of concept actually, and this will be more useful if we apply this in a vegetatively propagating crop. Say for example, potato. In those of you who work on potato seed certification is a very success story, and for a couple of viruses like 5 to 6 viruses, we do potato seed certification. So, why not just prepare a cocktail of the antibody? Just by one reaction you can say mother plant is free from virus or is not free. To do that, you need to start with what we did is, we stacked the coat protein gene of potato virus X and potato virus Y, and we found the system is working. Subsequently, we put the 4 virus coat protein gene of 4 different viruses together, and we found that the system is working. That means instead of doing 4 independent ELISAs, you do just one set of ELISA, and you can say that whether the mother plant is free from PVX, PVY, PVM and PVA. So, you are cutting down the cost. Remember, any cost you incur on any set of diagnostics has to be borne by your client, whether it is industry or whether it is farmer and so on and so forth. And this information again we published. So, that shows that instead of using just a simple ELISA, we can go for a simplex to multiplex ELISA. Okay? And that is what is called as multiplex ELISA. Now, besides multiplex ELISA, you must have gone through the PCR. Now, again in PCR, conventional PCR deals with only to detect only one virus at a time. Again, if you want to cut down the cost of PCR, the people have devised methods and what is popularly called as multiplex PCR. It could be a duplex PCR, it could be triplex PCR, quadriplex or multiplex PCR. Now, my colleague from Advanced Center for Plant Virology, Dr. Vikas Mandal and his group, they have been able to develop multiplex PCR for parallel detection of potato viruses. So, you can see that for large number of viruses, they have been able to develop multiplex. So, one reaction, you will be able to say whether the given sample has which virus and so on and so forth. Similarly, Dr. Banwal and his group, they have been able to develop multiplex PCR for garlic viruses. Similarly, again his group has been able to develop multiplex PCR for citrus viruses. And not only viruses, again there is a citrus greening bacterium disease which is a serious problem. So, he has been able to develop multiplex PCR for that too. Now, Having used these diagnostic techniques, molecular diagnostic techniques like ELISA and PCR, our group has been able to unfold the etiology of certain diseases, which I like to share with you. I don't know how many of you, those of you are working on plantation crop. Who is working on plantation crop? No. Okay, no. Large cardamom. In case of large cardamom, there has been two diseases, Chirke and Furke diseases. And the etiology was unknown till we worked actually on this. And when we started working on Furke disease, by using ELISA and PCR based diagnostics and subsequently cloning and sequencing, we were able to identify the virus, a new virus, what is called as large cardamom bushy dwarf virus, and this virus was very similar to banana bunchy top virus, and it was designated as a Babu virus. Similarly, as far as Chirke disease of large cardamom is concerned, we identified that there is a flexus virus associated with it, and it's a new flexus virus which was designated as Maclura virus, and this information was published in 2012 in Annals of Applied Biology. So what I am trying to convince you is that by using this molecular diagnostics technique, you can unfold the etiology of certain diseases which have not been established actually. And Chirke and Furke is one such example actually. Now, besides multiplex PCR or multiplex ELISA, if you have to detect multiple infection, there are certain other techniques which have also been developed in the recent past, and those of you, are conversant with the problems and going through the large number of papers, you will find that the technology what is known as microarray or the macroarray. These are the two nucleic acid based techniques which have the potential to detect large number of virus infection at a time. Okay? I'll just make a mention that USDA, United States Department of Agriculture, has developed a chip which is capable of detecting 52 viruses at a time. Just by developing a chip and using that chip, you are capable of, so what is called as universal plant virus chip by John Hammond from USDA. Nice. Using the similar approach, Dr. Barnwala and his group from Advanced Center for Plant Virology, they have also developed a chip what is known as ACPV chip, and this chip is capable of detecting the known viruses including the viroids actually. So, you need not to do independent reactions, just by using one chip you can be able to, likewise when this chip was used for grape, when the chip was used for chili, we were able to diagnose certain newer viruses or viroids actually. Then as I have said earlier, if you want to really know the unknown viruses, I mentioned the technique which is available now from 2009 onwards is the next generation sequencing or the deep sequencing. We also wanted to use the similar approach to unfold the etiology of urd bean leaf crinkle disease. Actually, those of you who work on legume viruses, or those of you who work with legume crops, they may be knowing, we knew this disease when Dr. Y.L. Nene and his group were there at Pantnagar, and we knew that it is a white fly transmitted, it's so on and so forth, but we did not know what is the causal agent actually. So then by using this deep sequencing coupled with RT-PCR and ELISA, revealed the association of a Carla virus, that is cowpea mild motor virus, and this information we published in 2015 in the journal Crop Protection actually. But still it is not very clear, because unless we really prove the pathogenicity, be careful, when you do deep sequencing or next generation sequencing, you are merely seeing the association of the causal agent. You are not sure whether it is the cause of the disease or not actually. Okay? So in order to be sure that it is the causal agent of the disease, we have to do the pathogenicity. Okay? So we have merely seen the association of the virus, but that does not mean that it is the cause of the disease. So that we are yet to confirm that would be in leaf crinkle is because of a Carla virus, that is cowpea mild mortar. So that's what I'm saying. Never be lured that ELISA and PCR and all these techniques are very powerful techniques. You can get away with bioassay. No. Bioassay is as important as ELISA or PCR. Because even if you do ELISA, you are merely seeing the presence of the protein. If you are doing nucleic acid based diagnostics, you are merely seeing the presence of the nucleic acid. But you are not checking the pathogenicity. You don't know infectivity. So, like ELISA and PCR, you have to perform bioassay to be sure that the given virus or fungi or the bacteria is the causal agent of the disease. So, bioassay is as important as doing ELISA or the PCR. As I said earlier, this information we published in 2014 in Crop Protection. Using this deep sequencing, again Dr. Barnwal and his group has gone for apple virome analysis. This technique is very powerful, particularly in case of perennial crops actually. And also let me tell you, when it comes to diagnostics, diagnostics is more relevant when it comes to perennial crops. It may not be as relevant in annual crops, because by the time you diagnose, the crop season is going to be over. But when it comes to perennial crops, if you can diagnose, then you have enough time to incorporate control measures and so on and so forth. So when we try to compare annual versus perennial, diagnostics has much more relevance in perennial crops like apple, citrus, and so on and so forth. So by using deep sequencing, his group has been able to see some of the grapevine leaf roll associated virus like 345, and then grapevine virus as AEF, he has been able to identify. This is how does the symptoms look like. Besides the diagnostic capabilities which we have developed over a period of time, if you really want to have a sustainable diagnostics, in order to have a sustainable diagnostics, both ICAR and DBT have developed programs like ICAR, Indian Council of Agricultural Research, had started diagnostic networks. They have also started diagnostic platforms actually. And in those platforms, IRI has been the main center, and subsequently we had the various ICR institutes and SAUs and so on and so forth. So it is always a good idea to have diagnostic network or the platform if you really want to have a sustainable diagnostics. Besides, DBT has recently started a program what is called as National Certification System for Tissue Culture Raised Plants. Now, in order to certify the tissue culture raised plants, for example, banana, for example, citrus, for example, many other crops, ornamentals and so on and so forth, the mother plants have to be seen that they are free from viruses actually. And in order to do that, the DBT has started a certification system for tissue culture raised plants. Those of you who want to have more information about this, you can visit DBT website and you will get more information. And under this program, Referral Center for Virus Testing of Tissue Culture Raised Plants is located at Advanced Center of Plant Virology, and then there are certain accredited laboratories around Delhi. Now, ultimate aim of diagnostics, howsoever the diagnostics you have done, but unless you apply these diagnostics for raising the virus-free mother plants or planting material, the diagnostic then is restricted to academic exercise only. Okay? I'll share with you some of the success stories which have come up as far as virus diagnostics is concerned actually. This is just to share with you a program what is called as Citrus Budwood Certification Program at Nagpur. Again, Central Institute of Citrus Research at Nagpur. And my colleague, Dr. Dilip Ghosh and his group has been able to develop this Budwood certification program as far as citrus is concerned actually. And by using, like we have potato seed certification program which has been developed by CPRI Shimla long back. So they have been able to develop, on the similar lines, United States Department of Agriculture has developed citrus budwood certification program. Just to share with you that during the last 10 years, 5 million disease-free plants developed at Nagpur were distributed to the farmers during the last 10 years. And this resulted in a resource generation of about 8 crores. And these plants, 5 million disease plants, have occupied approximately 11,000 hectare area in places like Andhra Pradesh, Gujarat, Karnataka, Madhya Pradesh, and so on. So, just to give you an example that how viral diagnostics have been very successfully deployed to raise. Similarly, I'll share with you banana tissue culture, which is a success story. This has been published by Dr. Barnwal and his group in 2019. It is a ICAR-IARI publication of Bank, Thailand. And if you go through this publication, which is available perhaps on ICAR-IARI website, you will find the farmers which have used virus-free banana plants for raising their crop, then in Maharashtra they have been able to raise a net profit of this much. Similarly, in Tamil Nadu, the farmers could have a benefit of about 3 lakhs per acre. Similarly, in Karnataka, they could have a benefit of around 4 lakhs per acre. So that is the kind of benefit you can have if you are using virus-free mother plant when it comes to banana. So that shows that virus diagnostics is relevant. It is useful for the stakeholder. Okay. Friends, as I was mentioning that there have been a considerable emphasis as far as virus diagnosis is concerned during the last 3 to 4 decades. But let us look at what is our preparedness. Despite the fact that we have been giving considerable emphasis, what is our preparedness? You will find that our preparedness is still tip of an iceberg actually. Let us look at some of the ornamental crops, including fruit crops like grape, then of course potato and so on. These are the number of viruses which have been recorded worldwide. But the fact remains, our preparedness is for a very few viruses or viroids and so on and so forth. Don't get worried about the absolute figures. The message which I want to give is that our preparedness is yet for a fewer viruses or viroids. That means a lot of work has to be done. That means you have lot of opportunities to work actually. Yeah. Much more has to be done. So you have a lot of opportunities to work, nothing. And let me share with you, in a recent study in 2019 by deep sequencing, it has been reported in 2019 that there are more than 100 million virus species on the Earth planet. Not just plants, it is on the Earth planet which includes everything. So that shows you have lots of scope to work actually. It's not that everything has been solved actually. There are certain major issues which need to be addressed. We need to have a dedicated facility for anti-serum production. We need to have a containment facility if you want to work on exotic pathogens. I will say pathogens. Even if exotic fungi are there or bacteria are there, we need to have, if you have to really have preparedness for them, we should have exotic containment facility. We should do virome analysis, and then of course, we should do on the spot grower friendly diagnostics, what is called as lateral flow assay or strip assay, as we do for in case of human beings and so on and so forth. I would like to share with you that a country like Thailand has a national facility for anti-serum production. Why not our country could also have a national facility for anti-serum production? Just to tell you that he is Professor Anupam Verma, and in fact he has been the architect of Advanced Center for Plant Virology, which is located at this campus actually. And he is a well-known virologist. Still he is with us as INSA senior scientist actually. This is just to give you an example that recent publications which have come up in relation to deep sequencing. These are some of the recent reviews with regards to phytodiagnostics, not only to viruses, but it includes everything, fungi, bacteria and so on. And the most recent has been in case of 2019, which is in Indian phytopathology, that is high throughput sequencing of plant virus discovery by deep sequencing actually. Okay? So these are some of the recent reviews. If you find time, you can have a look. I think this is my last slide. This is just to sum up, as far as the virus diagnosis is concerned. When we collect samples, we can either subject them to immuno-based diagnostics or we can subject them to nuclear-based diagnostics. Predominantly, when we talk of immuno-based diagnostic, we only talk about ELISA now. We don't talk about double diffusion or tube precipitin and so on and so forth. We only talk about ELISA for a single known virus or a multiple virus. Then when it comes to nuclear-based diagnostic, we either talk about PCR, microarray, and then of course, of late now we talk about next generation sequencing, and if you want to really know the unknown, novel viruses. This is just to share with you the Advanced Center of Plant Virology family. Dr. V.K. Banwal, who is the in charge of Advanced Center of Plant Virology. They are the members of Advanced Center of Plant Virology family. I mean, as I said, Dr. Anupam Verma, who has been the architect, and of course me, I was also associated. Even now I am associated as emeritus scientist. Thank you so much for your patient hearing.
So, any question? You people are lucky enough. When we were studying, we won't get such chance. Sir, we are really thankful, sir.
My pleasure. My pleasure.
And, gave us about all. Actually, horticulture and viruses, they cannot go separately. So we have to understand. Okay, if we want to do something, we have to understand this aspect. And sir, I know always you always blessed us with such a nice information. Sir, thank you so much from my behalf, from
my pleasure.
team.
My pleasure. And any one of you, if you wish to work on viruses, you can always write to Dr. Rashmi Agrawal, who is head plant pathology, and Dr. V.K. Banwal, and spend some time, maybe a week's time, and work with your sample. If you want to really analyze which virus or which fungi, which bacteria, you can always bank upon IARI.
Sir, this is a kind of
No.
Thank you so much. Thank you.
Thank you. Ah, yes.